So we've produced our 2 molecules of acetyl-CoA, and now each of those molecules of acetyl-CoA is going to enter what's known as the citric acid cycle. As its name implies, this is a cycle, so we're going to ultimately produce a product that is going to allow the cycle to continue endlessly, potentially. So we have this cycle, and each of the molecules of acetyl-CoA that we've produced are going to enter this cycle. So I'm going to draw the cycle for one molecule of acetyl-CoA. Understand that if we've generated 2 molecules of acetyl-CoA, each of them can enter this cycle. Now, acetyl-CoA is a 2-carbon compound. And when it is first introduced to the cycle, it attaches to a 4-carbon compound called oxaloacetate. By the end of the cycle, we need to remove 2 carbons from the combined 6-carbon compound called citrate that is formed by fusing acetyl-CoA to oxaloacetate. So when we introduce acetyl-CoA to the 4-carbon compound, we make a 6-carbon compound. We are going to eventually need to remove those 2 carbons again. So the first thing that's going to happen to the intermediates formed is we're going to reduce a molecule of NAD plus to NADH and a proton. Okay? The resulting compound is then going to lose 1 carbon in the form of carbon dioxide. So it loses a carboxyl group in the form of carbon dioxide. Okay? Then, as the cycle proceeds, we're going to again lose another carboxyl group and another carbon from the compound in the form of carbon dioxide. So here we see we have put 2 carbons into the 4-carbon compound. Now we have lost those 2 carbons in the form of carbon dioxide. The resulting intermediate, again, is going to reduce NAD plus to NADH. Okay, we're going to see CoA pop back in, because once we entered the cycle, coenzyme A was released from acetyl-CoA. It's going to come back into the cycle here, and ultimately form an intermediate where CoA will be replaced by a phosphate group, and through substrate level phosphorylation, we're going to generate a molecule of ATP from ADP. Okay? And that's actually through means of transferring the phosphate group first to GDP, which becomes GTP, and then ultimately we'll transfer that phosphate to ADP. Okay? Then we are going to reduce the molecule FAD to FADH2. Okay? So at this point in time in the cycle, we've removed the 2 carbons that we wanted to remove, and now what we're attempting to do is regenerate oxaloacetate, that 4-carbon compound that was here in the cycle that allowed us to attach acetyl-CoA initially to that molecule, forming our 6-carbon compound of citrate. So we're going to systematically now remove electrons, reducing FAD to FADH2 on our way to again generating that 4-carbon compound of oxaloacetate. And then there is a final oxidation and reduction event where NAD plus is again reduced to NADH and a proton. Okay? So NADH plus a proton. So this is the cycle. At this point in time, we've now generated oxaloacetate, which is our 4-carbon compound, and we can again repeat the cycle. So we've generated 2 molecules of acetyl-CoA in glycolysis. For each of those molecules of acetyl-CoA, we can carry out this cycle once, meaning that for a single molecule of acetyl-CoA, you will produce 3 molecules of NADH, 3 protons, 2 molecules of carbon dioxide, 1 molecule of ATP, and 1 molecule of FADH2. for 1 molecule of glucose, we generate 2 molecules of pyruvate, which means we generate 2 molecules of acetyl-CoA. So for 2 molecules of acetyl-CoA, we can double those numbers, which means for every 1 molecule of glucose that we split during glycolysis, by the time the pyruvates from that glucose are converted to acetyl-CoA, we can then produce a total of 6 NADH molecules, 6 protons, 4 carbon dioxide molecules, 2 ATPs, and 2 FADH2s in the citric acid cycle. So this is the citric acid cycle.